Acetyl coenzyme A, the mega molecule, the base of everything, pretty much everything that we sort of eat macronutrient-wise on a daily basis can be broken down into acetyl coenzyme A, or a few things that can produce acetyl coenzyme A, or a few things that acetyl coenzyme A actually goes on to sort of produce and end up combining with. So it also sort of still links to acetyl CoA in a way. And what's interesting is that while acetyl CoA, it seems like quite a big molecule, the actual acetyl part of acetyl CoA is the bit that sort of really matters. The coenzyme A part is sort of present in a load of things in the body, and it can be binded to things that aren't just acetyl. But the acetyl group is what the actual fatty acids, the proteins, the glucose, the carbohydrates, that's what they are sort of metabolized into, because coenzyme A is an absolutely massive molecule, and proteins and fatty acids are all sort of smaller when you consider chemical size on like such a small nanoscale compared to coenzyme A. And so it is the acetyl part that is the thing that these molecules are digested into. And the coenzyme is simply what makes acetyl CoA activated and allows it to be used in certain metabolic processes that are absolutely vital for the body to sort of function and exist, like the Krebs cycle and loads of other things that it is used to end up producing, things like cholesterol. Acetyl CoA is sort of the starting molecule of the sort of metabolism of cholesterol. And there's so many different things that acetyl CoA can be used to do that I myself don't even actually know and understand about yet. But what's interesting about the acetyl part of acetyl coenzyme A is that it is one hydrogen atom away from being a group 1 carcinogen known as acetaldehyde. And I have spoken about these two molecules having a lot of similarities before. But while they may be similar in chemical structure, their usefulness to the body is entirely different. Acetaldehyde is produced during the metabolism of ethanol, and it's a group 1 carcinogen, meaning it can cause cancer and is clinically trialed and proven to cause cancer inside of humans. It's not been tried and tested on animals or anything like that. It has literally been tried and tested inside of human, like species, human samples or whatever, and it can cause cancer, mutate your DNA, cause all the things that means that cancer can actually develop inside of your body. The fine lines of science and chemistry, the literal one hydrogen atom that separates acetyl from actually being acetaldehyde, means that your body can actually live and exist and function. And that's just an incredibly interesting thing to me. And I don't know, maybe you do feel the sort of exact same way. But the coenzyme A part of the acetyl molecule is sort of a very, very complicated thing. It's got a few sort of nitrogenous bases. I think it might have an adenine bit at the top. It's got a few sort of sugar rings. It's got a few sort of pentose ring thingies. It's got, I think, a few sulfur sort of molecules, which are very, very rare inside of the human body. And the sulfur bit is actually the thing that sort of binds to whatever is sort of on the end of the coenzyme A molecule. Because there are a few other things that coenzyme A can sort of bind to and activate that isn't just acetyl. Because while acetyl is sort of produced by a lot of the sort of foods that you eat, there are other things that sort of occur inside of the body that coenzyme A is actually needed sort of for. But what's interesting is that, as I said, so many of the foods and macronutrients that you eat can be produced and digested into acetyl CoA. Acetyl CoA is the reason that macronutrients actually exist and how calories actually exist. When fats, proteins, carbohydrates, and alcohols are broken down into acetyl CoA or intermediates, this means that acetyl CoA is essentially an energy storing molecule. A few of its bonds that need to be broken during the metabolic processes that acetyl CoA goes through, such as the Krebs cycle. And this means that acetyl CoA ends up yielding energy. It can produce ATP molecules. It can produce NADH molecules, which are then used to produce ATP during oxidative phosphorylation. The sort of acetyl CoA molecule is essentially the thing that is calorific. And that is why molecules like fatty acids, like glucose, like amino acids, all end up being metabolized to sort of form this molecule, or a few other things in the case of sort of amino acids and proteins, which I will get to in a little bit. But I have covered in the past the process of carbohydrates ending up into acetyl CoA, the process of glycolysis, the breakdown of glucose into pyruvate, which can then be turned into acetyl CoA. That is how carbohydrates can be turned into acetyl CoA, and that is why carbohydrates as a macronutrient are calorific, because acetyl CoA is an energy storing molecule. It holds energy in between its bonds, and when these bonds are broken during metabolic processes, it ends up releasing that energy in the form of ATP, and that ATP can be used to literally just make your muscles move. You can use it in your gym workout. It's a very sort of short-term molecule. it can only be stored, one molecule of ATP can be stored in your body for maybe 4 to 5 seconds at a time. That's how quickly these sort of processes are sort of going inside of your body. Carbohydrates produce, I think it's 4 or 3.75 calories per gram. I know proteins is either 4 or 3.75 calories per gram. I just can't off the top of my head remember which one's which in a weird sort of way. I've also covered the metabolism of alcohol into acetyl CoA. Alcohol, ethanol specifically, turns into acetaldehyde, turns into acetate, turns into acetyl CoA. That is how alcohols are actually calorific sort of substances. But even though alcohols aren't really present that much inside of a lot of the sort of stuff that you consume on a daily basis, it is still a calorific thing. And while I don't recommend consuming it to get your calories, because of the things that it does to your body, because of the carcinogens that it produces during its sort of way from alcohol to acetyl CoA, it's just another interesting thing that you may not have known before, that alcohols and the sort of thing that makes up alcohols, the hydroxyl group, and the way that that can then be turned and transformed into acetyl CoA is quite an interesting thing as well. But I haven't covered the metabolisms completely and entirely of fats and proteins into acetyl CoA. Fats and proteins are just like carbohydrates and alcohols, macronutrients. Fats have 9 calories per gram, proteins have 4 or 3.75 calories per gram, alcohols have 7 calories per gram consumed. All of this sort of stuff broken down into acetyl CoA, and the amount of acetyl CoA that these macronutrients produce when they are digested inside of the body ends up determining how many calories per gram that specific macronutrient actually contributes to. But now I am going to go through the metabolism of proteins as a sort of basic level as I specifically can, because proteins are actually a very, very complicated sort of base food group and molecule and chemical as a whole. Proteins are very long strands known as polypeptides of amino acids, and it's the amino acids that are actually the things that can be metabolized into acetyl CoA. When you eat maybe some meat or some beans or some pulses or whatever that contain a lot of protein, they will go into your stomach, they will go into your gut, and in these places they are digested by specific enzymes that break the bonds between the polypeptides that mean the amino acids can be separate themselves and roam like free as sort of amino acids essentially. And these amino acids then become small enough to actually be absorbed into the bloodstream, and once they're in the bloodstream they'll then be absorbed into cells where there are other enzymes, and these other enzymatic reactions will take place potentially in like the cytosol and cytoplasms of like all the cells inside of your body. But there are so many different types of amino acids that your body can sort of metabolize. Amino acid isn't like a specific thing that exists as one molecule. It's a group, it's a band of molecules that can sort of exist. Amino as a prefix essentially means nitrogen. Amino, amine, in sort of organic chemistry, means there is an amine group. It's got an NH2, NH3, NH1 group or whatever. And this means that all amino acids contain a nitrogen. But the reason they are an acid is because as well as having a nitrogen, they also have a carboxylic acid group, which is a COOH group. It's got a carbon bonded to an oxygen with a double bond, and then it's got an OH from that carbon. And the reason it's an acid is also a very complicated thing, because when it's in a solution, the sort of OH bit ends up sort of detaching from each other to make 2 separate ions, and that's essentially the thing that makes it a carboxylic acid. So that's the reason that an amino acid is called an amino acid. But there are 20 or so different types of amino acids that can be metabolized by the body. There's around 500 or more potentially in nature, but there's the amount that's sort of irrelevant to humans. But there are 9 of these that can't actually be produced by humans, because while a lot of these molecules can be metabolized into acetyl CoA or a few intermediates of acetyl CoA and a few of the metabolic processes that ends up going towards, the problem is that some of these processes can not sort of go backwards. A lot of the reactions that occur in the human body are reversible, and this means that from some sort of proteins they will go to acetyl CoA, and then if they lack that protein to be used in other areas, acetyl CoA can be used again to make that specific protein. But for 9 of these amino acids, this just doesn't happen. No sort of metabolic process inside of the body can actually be used to build its foundations into that specific amino acid. And so humans must obtain these 9 essential amino acids from the diet. So if we just consider the metabolisms of these amino acids into a few different molecules such as acetyl CoA, pyruvate, and a few other intermediates, these proteins can be classed into a few different categories based on what these molecules actually end up producing in the end. Now, some molecules can be produced into acetyl-CoA through being produced into pyruvate first, and these specific amino acids are alanine, serine, glycine, cysteine, threonine, and tryptophan. Some of these molecules during their metabolisms will not get turned into pyruvate first before they end up going to acetyl-CoA, so they just go straight into acetyl-CoA. And these molecules are isoleucine, lysine, leucine, phenylalanine, tryptophan, tyrosine, and threonine. Now, the reason that some of these molecules can end up appearing twice in some of these sort of categories is because they can have different metabolic pathways. For instance, tryptophan, threonine, or whatever the things that I said earlier, these can have two separate metabolic pathways where they can either go through pyruvate and be turned into pyruvate, or they can be turned straight up into acetyl-CoA. It just maybe depends or whatever, but they both have the sort of potential to go through those specific metabolic pathways. Now, there are a few other amino acids that can be turned into specifically Krebs cycle intermediates, and these are all the intermediates, all the sort of different molecules that are involved in the Krebs cycle: citrate, isocitrate, alpha-ketoglutarate. Then you maybe have citronil, citrate, fumarate, malate, and oxaloacetate. Some amino acids can be turned and metabolized into some of those molecules rather than acetyl-CoA themselves. And these molecules are glutamate, aspartic acid, asparagine, methionine, valine, tyrosine, phenylalanine, arginine, proline, histamine, and glutamine. And as I said, some can appear in both these categories because they may have separate metabolic pathways that can sort of either happen or either not happen. But there's also a different way you can categorize these proteins into ketogenic or glucogenic. Now, glucogenic and ketogenic essentially refer to glucose and acetyl-CoA. Now, a glucogenic amino acid means that it can turn into glucose without having to be converted into acetyl coenzyme A first. And the reason that glucogenic and ketogenic categories actually exist is because the link reaction between pyruvate and acetyl-CoA that I've talked about before isn't a reversible reaction. It can only happen from pyruvate to acetyl-CoA rather than maybe the whole entire glycolysis process where you can go from pyruvate to glucose and glucose to pyruvate. Once you get to pyruvate and you turn that pyruvate into acetyl-CoA, you can't go back to pyruvate, if that makes any sense. And that is why some amino acids can be glucogenic, ketogenic, but a ketogenic amino acid means it can be converted into acetyl-CoA. And some amino acids are only ketogenic, which means they can only be converted into acetyl-CoA. In the case of leucine and lysine, some can do both. Some can be converted into acetyl-CoA. Some can then be involved in Krebs cycle intermediates, because Krebs cycle, where it goes around the sort of cycle, then it turns into oxaloacetate, which can be involved in the sort of actual gluconeogenesis pathway. Pyruvate, which I've mentioned before, can turn into oxaloacetate, which can go up the gluconeogenesis pathway to make glucose, which is why Krebs cycle intermediates and the sort of molecules and proteins and amino acids that can be used to produce them, that's why they are glucogenic essentially. But leucine and lysine are the only two amino acids that are strictly ketogenic, simply because they can only be converted into acetyl-CoA. They can't be converted into pyruvate or any other intermediate or anything like that. Now, we've covered a lot on proteins in the last couple of minutes or so, but what about fats? Fats are a lot simpler molecules, thankfully. They don't contain any nitrogens. They don't contain any sort of other functional groups other than the carboxylic acid group, which I talked about earlier. The COOH group. A fatty acid must have this to be a organic fatty acid, and a fatty acid occurs when the COH bit of the carboxylic acid can sort of split when it's in solution, and then you have a carboxylate ion as well as an H+ ion, and that would end up meaning that it's acidic. Carboxylic acids are often quite weak acids compared to inorganic acids like hydrochloric acid or sulfuric acid, or any of that sort of really nasty stuff which you don't want inside of your bodies whatsoever. But carboxylic acids are a lot weaker, which is why they can sort of be metabolized into fatty acids and consumed in those sort of forms. But fats and fatty acids specifically are metabolized into acetyl-CoA through a process known as beta oxidation. The acetyl part of acetyl-CoA has 2 carbons in it, and that is incredibly important. The acetyl-CoA, obviously you need 2 carbons. The CoA molecule is waiting for that to have an acetyl group, attached onto it. It needs 2 carbons plus the sort of oxygen double bond or whatever, and beta oxidation of fatty acids is how the acetyl-CoA molecule actually achieves that. Beta oxidation, in a summary, ends up cleaving 2 carbons off the end of the fatty acid chain and doing a few things to that, or actually before it sort of cleaves it off the molecule, and that will mean that it can be converted into acetyl-CoA. And the first step of beta oxidation is adding a fatty acid to a coenzyme A molecule to make an acyl CoA molecule. Now, an acyl CoA molecule is sort of dependent. It's not sort of strictly a molecule. It can have any sort of length chain of fatty acid on that sort of CoA molecule. Acyl is just a way to describe a fatty acid attached to a coenzyme A molecule, and that's the first step. It simply replaces the OH of the carboxylic acid with the sort of coenzyme A bit, which is a very complicated molecule, which I'm not going to explain any more about because that's probably a video for another time. And the second step of beta oxidation is an acyl CoA molecule plus an FAD molecule ends up turning into a trans-enol CoA molecule plus an FADH2 molecule, and this reaction is catalyzed by the enzyme Acetyl CoA dehydrogenase. And the third step of beta oxidation is the trans-enol CoA molecule ends up being added to a water molecule, and the spontaneous reaction occurs without an enzyme needed into hydroxyacyl coenzyme A. And then you take that hydroxyacyl CoA molecule and you add it to an NAD plus molecule, and that will end up turning into NADH plus ketoacyl coenzyme A, and this reaction is catalyzed by the enzyme hydroxyacyl CoA dehydrogenase. And in the final step of beta oxidation, that ketoacyl CoA molecule is added to a separate coenzyme A, and this will end up turning into acyl CoA plus the acetyl CoA that our body so sorely needs to actually run its metabolic processes. And this reaction is catalyzed by the enzyme thiolase, and that will end up continuing. The acyl CoA that was producing the final reaction will go through those steps again and again and again until you sort of got to the end of the chain, and that's when it's done. The problem is that it's never as simple as that, because that only applies to saturated, even-numbered fatty acid chains. It's a very sort of specific need for your body to have for it to actually go through that specific process that is so easy to explain and so easy for your body to handle. When double bonds and unsaturated sort of fatty acids get involved, as well as odd-numbered fatty acids, it gets a lot more complicated, but I still am going to go through that in this video. Now, for odd-numbered saturated fatty acids, which means it has no double bonds inside of its hydrocarbon chain, you just simply, in the last reaction step, when it gets right to the end of the fatty acid chain, it ends up producing a propionyl CoA molecule, as well as an acetyl CoA molecule, and that propionyl CoA molecule will end up being metabolized into succinyl CoA, which actually is a metabolite and an intermediate inside of the Krebs cycle, which will then end up going through its processes, turned into succinate, turned into a few other things, and then turns into oxaloacetate, and then you end up getting energy from this continuous cycle occurring inside of your mitochondria. So for odd-numbered saturated fatty acids, the end is still okay, but what happens when a double bond ends up being involved, which can seriously scupper with the sort of stage where you get to the sort of having to cleave off the 2 carbon molecules, but you can't because there's like a double bond there, and it all ends up getting a bit confusing. How exactly does your body handle this? Because sometimes the acyl CoA dehydrogenase enzyme simply won't work if the sort of acyl CoA molecule, the bit at the end where there's sort of the 2 carbons, already has a double bond, because the trans-enol CoA molecule actually has a double bond. That reaction ends up producing a double bond onto the trans-enol CoA molecule. But the problem is that if there's already a double bond there, that will scupper it, because it might not be a trans double bond, it could be a cis double bond, and that's going to produce a lot of problems. And so you can use a few other enzymes, such as enoyl CoA isomerase, which may turn it into a trans double bond instead of a cis double bond. Or your body may have to call in the help of 2,4-dienyl CoA reductase to solve the issue. And boom, beta oxidation is complete for all the sort of variations of fatty acids that exist. Interestingly enough, saturated fatty acids are the easiest for your body to digest, which is quite interesting considering back in the sort of olden days, the 1900s, there was a big craze going around where people were saying that saturated fatty acids were absolutely impossible to digest and couldn't be absorbed, couldn't be metabolized, And this ended up leading to the rise of trans fats, hydrogenation processes, and all that stuff that's absolutely horrible for your body and your heart and everything in between, to be honest. But now we have a more advanced understanding of science. Even people like me, someone who's quite young, a 16-year-old, is able to access stuff on the internet, that is able to educate themselves on their diet, on their health, and be able to improve themselves for the better. And you can do the exact same thing by going on ChatGPT or whatever, doing your research on diet, doing your research on your own sort of specific dietary needs, and a whole bunch of other stuff, which I'm sure you will find very interesting. So I wish you good luck on your self-improvement journey. I hope you enjoyed this long video on acetyl CoA and a bit of a summary on how it can be produced by glucose, alcohols, fats and proteins, and why acetyl CoA essentially means that these molecules are actually calorific, which is an insane sort of revelation that you may not have realized before. I'll probably do individual videos that go into greater depth into how each and every sort of amino acid can be metabolized into acetyl CoA, or sort of the other molecules that it can be metabolized into. For instance, tryptophan, I've done a video on that metabolism. I've done a video on its metabolism to vitamin B3, because of amino acid tryptophan can be produced to vitamin B3, but it can also be produced into acetyl CoA. It can also be produced into pyruvate, which can also not be used to produce acetyl CoA. It's all a very interesting thing. Everything in the body is absolutely insanely sort of complicated, but that's what makes it so interesting, because it's all happening at such an insane rate that you couldn't even sort of understand it if you sliced off like a cell, a single cell. Every time you like rub your sort of arm or something, loads and loads of dead cells just end up falling off your skin. Every time you sort of brush your arm or something. It's, yeah, I personally really like science. I hope you can kind of resonate with me on that in any way. If you don't, maybe you still learned something new. So with that, I hope you enjoyed, and I highly recommend subscribing, because I love this community of young tribal people who are willing to improve themselves